Okay, so we've talked about the control center of the cell, and now we're going to talk about the organelles that build proteins. And what I'm going to do is I'm going to go straight through the PowerPoint, and then I will put up the corresponding videos that are in here so that you can watch them and answer those questions. So, what organelles help make and transport proteins and other macromolecules? So here we got to understand what is meant by macromolecules. In our next part of the lesson coming up, we are going to talk about the chemistry of life. And we will talk about four main macromolecules that we need. And macro simply means very large molecules. And so in cells, proteins do a lot. And usually when we think of proteins, we think about muscles. But proteins do a lot more. They send chemical messages. They aid in structure and support. You have gate proteins in your cell membrane. Proteins function as enzymes, hormones, and a whole bunch of other things. And so we will probably talk a lot about proteins as we go along. So the first organelle that we're going to talk about is the ribosome. And so the ribosomes, if you remember from the nucleus, are packaged together in the nucleolus. Ribosomes don't have a membrane, so they are found in both prokaryotes and eukaryotes. And ribosomes are associated with a nucleic acid called RNA, ribonucleic acid. And we will talk about the role of RNA and ribosomes in making DNA and carrying that secret code information that we have locked up in the nucleus out into the cell. And so they are important not only for protein synthesis, but also for us passing on our genetic information to all the cells in our body and also to our offspring. So attached to the nuclear membrane is an organelle called the endoplasmic reticulum. And so there's two types of endoplasmic reticulum, and they both have two different functions. We have the rough endoplasmic reticulum, and it's called rough because inside of its folds, it has ribosomes embedded inside it. And so it is rough with ribosomes. So the endoplasmic reticulum is kind of a place where proteins get put together, added together, amino acids are being put together. And so it's kind of a little factory of making proteins. Okay, so I will play this video for you in the formative. I found out that when I record these videos like this, it doesn't pick up the sound in the videos. So after we get done here, I will have those posted in the formative for you. Okay. The other part of the endoplasmic reticulum is called the smooth endoplasmic reticulum because it doesn't have any ribosomes. And it has a different function than the rough endoplasmic reticulum. It produces lipids and carbohydrates, and it takes part in the detoxification of cells. And so anytime there is something that's not supposed to be there, it's considered a poison. So it is the place where poisons get worked on. So we can then get rid of them out of our system. So here is question number 6 to put in your notes about the rough endoplasmic reticulum. Again, I'll play this coming up. Here are your two questions about the smooth endoplasmic reticulum. So you put those on your notes page. You can either use a piece of notebook paper or that graph paper that's the end of each section of the lesson. And so then we have the Golgi apparatus. It's also known as the Golgi complex. So you'll see it written either way. And you'll see it on test questions from now until when you graduate from college. Either way, Golgi apparatus or the Golgi complex. And here are your two questions, 9 and 10, to answer. And I will be putting those videos following this video. Okay, so here we have a little synopsis of what's going on when proteins are made. So inside the nucleus is where we have the genetic information. We have it protected in the nucleus. And so in the center of the nucleus is an area called the nucleolus, where ribosomes are packaged together. And remember that ribosomes do not have a membrane around them. Surrounding the nucleolus is our chromatin, which is all of our stringy DNA hanging out. We have 23 different colors of string for humans, but we have we have one set from our mom and one set from our dad. So we actually have 46 total chromosomes in our nucleus. So if we were going with colors, we'd have 2 red and 2 purple and 2 aquamarine. Okay? And so this has the information about how to build proteins, what proteins we need to make, how many freckles you're going to have, how tall you're going to be, all that good stuff is here in the chromatin. But that message doesn't leave the nucleus. So the DNA doesn't go out here into the cytoplasm and say, hey, make this protein. It stays protected. So RNA, ribosomal nucleic acid, takes that message out of the nucleus and into the rough endoplasmic reticulum. So remember that the rough endoplasmic reticulum is called rough because it has ribosomes associated with it. So those proteins get made and they can either be pinched into a vesicle. A vesicle is simply a little taxi cab that's going to take something we want to protect. We don't want it to get damaged and take it to where it needs to be. So it could be to another organelle, or it could be outside of the cell itself. Or it could go to the smooth endoplasmic reticulum also. So proteins are assembled on the ribosomes. Remember, those ribosomes are embedded in here in this rough endoplasmic reticulum. So some of the proteins are completed. They've been folded the right way, have the right 3D shape, and they can be sent off their merry way to do the job that they're supposed to. Or they can have some more processing that needs to be done. So the proteins that need some more processing end up going to the Golgi apparatus or the Golgi complex. So when the vesicle carries that protein, and the reason why the vesicle carries the protein is because out here in the cytoplasm, it could be the wrong pH, it could bump into another molecule and get damaged. We could have some enzymes that break it down. So encapsulating it in this little taxicab or vesicle helps protect it until it gets to the Golgi apparatus. So the vesicle fuses here with the Golgi apparatus, and that protein gets packaged and reassembled and polished to the way it needs to be to do its specific function. And so this is where the protein gets wrapped and folded and painted and ribbons put on before it's ready to be sent out in another taxicab to do its job. So here's a breakdown of one whole big picture of what we just talked about. Ribosomes made. Endoplasmic reticulum is rough here because it has ribosomes associated with it. And the ribosomes aid in protein synthesis. And they are associated with ribonucleic acid. And so the message for what protein to make and how many freckles you're going to have and how long your hair is going to be, that sort of thing, that message is kept in the nucleus, locked up in the nucleus. But RNA carries that information out of those nuclear pores into the rough reticulum to where the proteins are starting to be manufactured. So these proteins are long chains of amino acids, and those long chains, think of it a long chain of beads, end up getting folded and twisted and put into a certain 3D shape. Each protein has a very specific shape, and if it gets broken or damaged in any way, it won't be able to complete its task. And so it gets protected in a little taxicab called a vesicle, and remember that the smooth endoplasmic reticulum also synthesizes carbohydrates, detoxifies, is a detoxifying area, and synthesizes lipids. And so this little package gets sent off in its little vesicles, little taxicab, and it gets sent to the Golgi apparatus. And in the Golgi apparatus, the protein further gets polished. It gets more folding. It gets spot checked. It's like it's getting all wrapped up and a bow put on it so that it can go out and be used out into the cell. And so the cell can actually send it to another organelle, or it can it can have it exit the cell and be taken to another cell to do a different